PSSM1. What is it? PSSM, or polysaccharide storage myopathy, is a hereditary condition that can create muscle damage in horses. There are two types of PSSM. The first, PSSM1, is caused by a mutation in the G1S1 glycogen synthase 1 gene that changes the sequence of amino acids. The second, PSSM2, is a form of abnormal polysaccharide storage that is not caused by mutation in the G1S1 gene, and the cause of this type is still undetermined.
The function and operation of the muscle. When a horse that does not have the G1S1 gene mutation exercises, the normal function of the muscle will be much different than that of a horse with the mutation. In a normal functioning muscle ATP, adenosine triphosphate is used to stimulate muscle contraction. Because the amount of ATP stored in the muscle is so small, it must use other pathways to produce more ATP when exercised. There are several different ways to produce ATP through these pathways, but the most prominent pathway is through the production of glycogen by the breakdown of glucose molecules obtained in the horse's diet. Once glycogen has been used by the muscle for energy, it produces end products that are left in the muscle called lactate and pyruvate, which leads to muscle fatigue after a hard workout. This is the cause of the stiffness you feel in your body after a leg day at the gym. After the glycogen has been used for energy and its end products are left behind, the muscle then burns the end products into CO2 and leaves the body through the respiratory system. Where does the G1S1 gene come in? When glucose is first obtained into the body through the horse's diet, mainly through forages, it is fermented by bacteria which then is produced into glucose. Once glucose is synthesized into the body, the G1S1 gene then catalyzes the addition of glucose monomers to the growing glycogen molecule through the formation of alpha-1 4-glycoside linkages. To sum this up, the G1S1 gene is responsible for the formation of glucose molecules to the final end product, glycogen. How does this relate to PSSM? In a horse with PSSM type 1, the GYS1 gene has a mutation in its amino acid sequence, a single base pair substitution. Because of this mutation in the gene, the amino acid codons that would normally read as a stop codon are changed. Therefore, this mutation results in the tRNA to continue reading the amino acid sequence, and the production of glycogen increases, even though glycogen is no longer needed as an energy source by the muscle. Once the body stops exercising, the glycogen continues to store in the muscle even though the energy is no longer needed, and as a result of this, the horse begins to show symptoms such as muscle cramping, stiffness, reluctance to move, sweating, and muscle weakness, otherwise known as tying up. How is this mutation transferred? PSSM1 is an autosomal hereditary condition that is either dominant or recessive depending on the species. For instance, with pigs, this mutation is autosomal recessive, but in the case of horses, this genetic mutation is autosomal dominant. When a gene is autosomal dominant, that means that the offspring needs at least one of the mutated genes from their parents to be affected. How to test for it. In order to determine if your horse has PSSM alleles in their DNA, you can get samples from the roots of the horse's mane or tail and send them to Animal Genetics. However, even if a horse tests as negative for the mutation, they can still have PSSM because a test only tests for the GYS1 mutation. Horses with type 2 do not have a mutated GYS1 gene, therefore a test for this type is impossible. The most common breeds for this genetic mutation are Percherons and Belgians.